for sensory information in the environment, say, let's just say, this is my second favorite stuffy. That's my neuron stuffy. So how many neurons does it take for the sensation of me feeling the stuffy, what it feels like, the touch sensation, to get to my cortex? Does anyone know? We haven't talked about this yet. This is all new. Any guesses? 1 million, 5 million, 2,
10,000.
10,000. Believe it or not, it is a 3-neuron system. Did you know that, Sydney? Yes? Did you know that, or was that a guess? You knew that. That was a guess. Okay. It is a 3-neuron system. We're going to talk more about that when we get to the spinal cord part of the course as well. It's not sensory information. So it's just a 3-order system. 3 neurons. First order neuron, second order neuron, third order neuron. We'll learn about that. That's sensory. That's somatosensory information coming from the environment. To get to my brain takes 3 neurons. Okay. How about motor? Motor starts in my which part of the brain does it start? Precentral gyrus, right? That's where the cell body is going to be, that gray matter we talked about. How many neurons in sequence does it take to get motor information out to the muscle? Starting from here. Does anyone know?
4.
No? Close.
5.
2. 2. And this is going to be, you're going to hear this over and over. You're going to hear it again. This is a lot this year. You're going to hear it a whole lot more next year, too. We're going to talk about the 2 neurons in sequence. There's the upper motor neuron. That starts up here in the precentral gyrus. And then we're going to talk about the lower motor neuron. The lower motor neuron actually exists in the ventral brake horn of the spinal cord. We'll talk about that. But amazingly enough, somatosensory information, 3 neurons in chain. And motor information, just 2. Okay. All right. I'm thinking about parts of the neuron. I'm going to have my neuron stuffy right here. Does anyone know the parts of the neuron? Did somebody say soma? Axon. Okay. This guy's going to be the axon here. Okay. These are your dendrites up here. This is an example of a multipolar neuron. We're going to talk about that later. But these are your dendrites. In general, your dendrites are going to be attached to your cell body or your soma. That's this part here where the eyes are. Okay. Below that, there's axon hillock. We're going to talk about that. And then you have your axon. At the base of your axon, you have something known as your axon terminal. Okay, all the basic parts. And then, if you notice, the axon here has there's some puffy parts in the middle, and there's striations. Yeah, good. That's going to be your myelin. So your myelin is going to be your puffy parts, and we'll talk about what these striations are called. But this, sorry, did somebody have a comment? No? It's fine? That's okay. You can talk. Feel free. Feel free to answer questions. That's great. But in general, this guy has all the information that you need to know about parts of the neuron. Now let's go through and start labeling here. Start with parts of the neuron. And if I said that the environment, these are my black markers. So let's say that the environment is here. Okay? And then, given the environment is there, the flow of information, the flow of, let's just start with somatosensory information, is going to go from the environment. So it's going to come from the top part of the neuron, and it's going to flow in this direction to the next neuron in sequence. Okay, so I'm going to say this is the flow of information. Okay, so the flow of information is going to come from the environment here down through this neuron to the next neuron in sequence, and then this goes to the CNS. Everybody got it? So sensory information is coming in. Now, when we think about parts of the neuron, and I just showed my little stuffy, there are these things up that are going to pick up information from the environment. Which part of the neuron would this be? Yeah, these are your dendrites. Good. D-E-N-D-R-I-T-E-S. Those are your dendrites. Okay, they're going to receive information from the environment. And then underneath your dendrites, you have here, this is going to be your cell body. We talked a little bit about before. Okay, so shown in blue there is the cell body. Another name for, does anybody know another name for a cell body? It's a soma. Okay, it's going to be your soma or your cell body. So let's go ahead and label this bluish color, the darker blue. It's going to be your cell body. Your cell body, this is nothing more than the nucleus of the cell body. And then moving on down the chain, we have this region of the neuron here. It's a connection, kind of a connector piece. Does anyone know what that's called, that part? It's going to be important for detecting what's called threshold. Dr. Blair said you started a little bit with action potentials, just a squeak. Say, I heard it. Axon hillock, yes. That's going to be your axon hillock. We'll talk about what that does here in a little bit. That's going to be your axon hillock. Your axon hillock is nothing more than this connector piece between the cell body and then the part right under your axon hillock is going to be your axon. So I'll put the axon here in red. This is nothing more than the basic parts of the neuron. Okay, shown in red here is going to be your axon. At the very bottom of the axon, I have this part. It has a special name. It's called your axon terminal. Let me put that in a different color. Maybe I'll do that in brown. This is going to be your axon terminal here in brown. So that's the endmost portion of the axon. So flow of information goes from the environment, perceived by the dendrites. Okay, that flow of information is transmitted through the axon hillock, through the axon, something called an action potential. We'll look at that in a minute. And then finally it terminates here at the axon terminal. There's going to be a space. So this is the first neuron in sequence. Okay, there's going to be a space here. And I'm going to just show this space in this green color. This is nothing more than the space between this first neuron and the second neuron. So here, this space has a special name. It's called a synaptic, S-Y-N-A-P-T-I-C, cleft, C-L-E-F-T. So that's going to be your synaptic cleft. And your synaptic cleft is nothing more than that space between neurons. Okay, so everybody see where we are? So the flow of information comes from the environment through this first neuron and then has to somehow traverse through that space to provide the information to the second neuron in sequence. Since this first, I've called it the first neuron. Here I'll put, this is the first neuron in sequence. Okay. This first neuron, since it exists before the synaptic cleft or before the synapse between neurons, it's called a presynaptic neuron. Okay. And that means that this neuron here, put it over here, it's the second neuron in sequence. This is going to be your postsynaptic neuron. Yes.
How does that work? Really aren't all I'm sorry, all of what? All of the neurons.
So the space is the space between the first neuron and the second neuron, or the third neuron. Now, what's your question?
Why is one presynaptic? The other one's going to be in front of another neuron, right?
Okay, I understand what you're saying. So the presynaptic neuron is thinking about the flow of information. So you're going from your first order, so think about somatosensory information, your first order neuron to your second order neuron. Your first order neuron is going to be presynaptic, and then the second order neuron is going to be postsynaptic. But then it's going to flip. Once that action potential goes through in the three neuron system, that's going to be your presynaptic. If there's another synapse here, remember it's only a three neuron system. Okay? In motor, there's only two neurons. So the presynaptic neuron is going to be up here, but the post is going to be down below. So anytime you're in front of or behind the synapse, that makes a difference.
I was going to add that in the case of what Dr. Bartlett is describing there, the preganglionic neuron, that first neuron in the sequence, is actually embedded in the skin. So there's no neuron in front of it in that case.
Yeah. So does that make sense? Any other questions? Everybody got it? In thinking about if we labeled all these parts over here with the dendrites of the presynaptic neuron, this postsynaptic neuron also has the same basic parts. Okay, so in other words, these would be the dendrites. I'll just keep them the same color. These are the dendrites of the postsynaptic neuron. And they'd be responsible for receiving information from the presynaptic neuron. Okay. And then you have your cell body, same thing. Okay. Did you have a question, Avery, or just stretching? Stretching? Okay. Okay. So in general, those are going to be parts of the neuron. And let's move on over to the next piece. And that's going to be looking at an action potential. Okay, the action potential is going to talk about how the signal traverses from essentially the environment. We'll start with the environmental piece, so mechanically gated channels, through the axon terminal. Okay? Activate that first neuron in sequence. And Dr. Blair, did you draw the action potential yet? Or
No, we didn't.
Haven't drawn this? Okay, so this is going to be a little bit new. Has anyone seen the action potential graphs before? Yeah, a few people? Okay. Anybody want to come up here and draw it? Bummer. Okay, so starting out, if I can ask some questions then if you have seen this before. If I had to give minus 70 a name, does anyone know? And we're going to start here on the y-axis. We have the membrane potential in millivolts. Okay, so I go from minus 90, and I get more positive as I go up to plus 10. Okay, and I can tell you we're going to start our time zero here at minus 70 millivolts. Does anybody know what that minus 70 or where we're starting at? See, you know it. Great. Okay, I'm going to label that number 1. Okay, put it there. And over here, I'm going to say number 1. You told me minus 70 is my resting membrane potential. Okay. Everybody got that? So pretty basic stuff. So now, the best way I know to describe this is to give you an example. They're actually what are called mechanically gated channels. Okay, in the dendrites. Mechanically gated channels live in the dendrites, and that would be in the periphery. So you have, let's say, a pin. Okay, if you get poked with a pin, you're going to feel the pain, right? So let's say you take a pin and you put it up to your fingertip. You feel it, but it doesn't hurt. Right? Has everybody had that experience before where you just touch it and it doesn't really hurt yet unless you push harder? Okay? So let's say I started with a pin and I just pushed it into my finger, but it doesn't really hurt yet. Okay? So that's going to be represented here. Okay? I'm going to poke with a pin, but it doesn't hurt. I don't feel the pain. Okay? So what's happening is there's some local mechanically gated channels that are being activated by the pressure of the pin, and that causes sodium to come into the cell a little bit, but we haven't reached minus 50 yet. Does anyone know what minus 50 represents? So this is all or nothing. Yeah, that's going to be your threshold, right? So that's going to be the amount of sodium that's rushing in. It didn't change the membrane potential of the neuron quite enough to reach that threshold. So we're going to label number 2 here. And the thought is that I have a pin and I'm just touching my finger, but I don't feel pain yet. It's not quite there. So that's going to be a local potential. And your local potential is going to be mechanically gated channel. So in other words, that sensation of pressure causes opening of the channel to allow, the mechanically gated channel, to allow sodium to start rushing into the cell, but it's not quite hard enough to reach minus 50, which was going to be your threshold. That's going to be your all or nothing point. Does that make sense? Yes.
Mechanically gated.
Gated, yeah. It's a mechanically gated channel, and I'm just using pain. There's lots of mechanically gated, but that sensation of pain. Okay. Now let's say, so you didn't quite make it with the first attempt, but the second attempt, yep, Valerie. Imagine a channel that is on the outside of the cell. So you have a cell, you have your cell membrane, and you have a channel here in the middle of the membrane. Okay? That channel has a little gate on the bottom, a swim neck. Okay? So that gate opens whenever I apply pressure to my skin, and it opens a little bit and allows sodium to rush in. Well, sodium comes in, then I start to become more positive. Okay? In order for this mechanically gated channel to allow enough sodium, when it allows enough sodium in to cross threshold, then you're going to get your action potential, which is the voltage gated channel. So your mechanically gated is due to some sort of pressure, pain, temperature, proprioception, anything, any kind of sensory, some sort of sensory system that's linked to the channel to allow sodium to enter into the cell. Does that help? Okay. That makes sense to everybody? Okay, that's going to be your mechanically gated channel. Lots of different modalities to get those channels to open. Okay, second time, I take my pin and I push, and then I say ow. Okay? When I say ow, that means that signal has actually been transmitted. And what happens there is I go from this local potential back down to resting membrane potential, and I'm going to go up past the -50. That means that enough sodium has entered into the cell to increase the membrane potential, make it more positive. Okay. Once we hit this -50, we're going to go all the way up, and that's the action potential. Okay. When you reach threshold, you go from mechanically gated channels to an all or nothing phenomenon known as your action potential, and that is sodium starts rushing into the cell. Okay, sodium starts rushing into the cell, and that's a voltage gated. So this is number 3. It's going to be right here at the point where we reach -50. That's called threshold. Okay, and at threshold, enough sodium has entered the cell to cross that -50 millivolts, and voltage gated channels open, specifically sodium channels. Okay, so your sodium channels open? Yes, Jonathan.
One more time how you said when it crossed the sodium started. Can you repeat that part?
Yes, I can
Like
try.
what
Tell me if I
thresh-
say it right. So
like
tell
what
me
the
if
threshold is into that.
Sure. So, let's go back. Maybe I'll go back to this picture real quick. Okay. Let's imagine that, maybe if I explain it with a picture as opposed to the graph, it might be a little bit easier. So imagine that the dendrites have this mechanically gated channel, okay? And the mechanically gated channel that lives here causes the influx of sodium into the cell, okay? And as the more pressure you get and the more sodium you get, it builds up here. But once it reaches the axon hillock, that's where you get the once you get to the -50, then it crosses the axon hillock and causes the action potential to go down the axon. Does that help? Is that what you wanted to know?
So the dendrites are
The mechanically gated channels are located in the dendrites. I was going to get to this in a minute. I was just trying to go through the graph first, but that's okay. I'm just trying to explain it as best I can so that you understand it. So in the dendrites themselves of the presynaptic neuron that's associated with the environment, okay, attached to my skin, I have those mechanically gated channels. Those mechanically gated channels they can be mechanically gated by a lot of different modalities. And the example I'm giving is just pain. Okay, when do you feel the pain? When it actually transmits up. Does that make sense?
Yes.
Okay. I think maybe the next one will help. But let's go through. So this is just looking at a basic action potential. Okay. For the basic action potential, so here at this point here, and I can label that. I'll label that number 4. Okay. At time point number 4, sodium channels are still open. Okay, and sodium is rushing into the cell to make the cell more positive. Yes, Alex?
Can we also call that depolarization?
Yes. Yes, that's the depolarization, going past threshold. That's when it's depolarized. Yeah.
Okay. So
Try not to use words I haven't said yet, but you got it. If you're depolarization, we're going to talk about that in a minute. That's fine. Yeah, depolarization versus hyperpolarization. We're going to get there. Okay, that's going to be when the neuron depolarizes. Okay, that means it fires. That's the action potential. Okay, so sodium rushes into the cell, and then here at this point number 5, sodium channels close. Okay, and potassium channels open, and to make the cell more negative, the potassium is going to leave the cell. Okay, so that's 0.5, and here this is going to be, you're going to come back down as potassium leaves the cell. You're going to come back past threshold, and you're actually going to get, we already talked about the word a little bit, this depolarization. You're going to get a hyperpolarization. Okay, and that's going to go below resting membrane potential. Okay, where potassium channels are still open. Potassium is still leaching out of the cell. And then eventually, potassium channels close. Let's say here, 0.6. Say potassium channels close. Okay, and as potassium channels close, you get back to your resting membrane potential. Okay? Questions? This is just these are the basics of an action potential. And thinking about it, when the cell reaches threshold at 0.3, that's going to cause a depolarization. And when the cell membrane potential is below the resting membrane potential, that's a hyperpolarization. Okay, so here you fire, here it's even harder to fire because you're so much below the resting membrane potential. Questions? So, did you have a question? Oh, Sydney?
Can you say number 6?
Yeah, 6 is going to be where, so 5 is where your potassium channels are going to start to open. So you're rapidly losing potassium out of the cell here. I'll write that down. And you keep losing potassium so much so that you go below the resting membrane potential, and that's going to be your hyperpolarization. Okay. At number 6, at this point, then the potassium channels close so that you come back to your resting membrane potential. Okay, and I'll put here, this is potassium channels open. Yes, Avery? No, you're just stretching. Okay, gotcha. Okay, this is just a basic action potential. What I like to do is go from this picture and then go back to the next synaptic communication and talk about what that means for the two neurons in sequence. Okay, and this is, Jonathan was asking me, so I went back a little bit, but now I'm going to go forward, and this is where we need to be right now. Okay. So like we mentioned before, this is going to be the two neurons in sequence. We're going to get information from our environment. And as I said in the dendrites, I think the dendrites were this blue. So this kind of blue here is going to be my dendrites. Okay. From dendrites, you're going to have, in your dendrites, are going to exist your mechanically gated channels. Okay. Mechanically gated channels are there, and the example that I use is pain. So inside the soma, you're going to have sodium, bless you, sodium's going to rush into the cell. Okay, Okay, when the mechanically gated channels are activated, it's going to cause sodium to rush into the cell. And when the inside of the cell reaches minus 50 millivolts, which is threshold, okay, and that threshold, look at what color I used, that threshold is going to result in the action potential reaching the axon hillock and transmitting down the axon. So for the action potential, this all or none process, once we reach minus 50 millivolts, is just going to go. We're going to have action potential traveling, and the action potential is just going to be your voltage gated ion channels are going to open. So this is your action potential. And the action potential is where your voltage gated channels, these are voltage gated sodium channels, open. Okay, and that's going to transmit all the way down the axon. Yes? On the axon, yes, on the axon. The dendrites are mechanically gated. So once we reach the axon hillock with a signal of or a membrane potential of minus 50 millivolts, then that's going to activate your voltage gated channels. Once your voltage gated channels open, it's a continuation all the way down the axon to the axon terminal. Yes, Grace. Yes, those are great potentials. And that's what I was trying to demonstrate that in the beginning by poking yourself with a thing that you couldn't quite feel it yet, so you didn't reach threshold. So that's going to be your graded local potential. Okay. Yes? Gabby.
Action potential.
Action potential, your action potential is going to be the opening of your voltage gated sodium channels. So your voltage gated sodium channels are going to open. Okay, that's going to represent that action potential that we just saw on that graph. We're going to go straight on up to the top. Okay, that's the firing. And action potential is an all or none phenomenon, whereas the local graded potentials of not quite feeling the pin on the outside from the environment, that's going to be your local graded potential, and it doesn't always have to go. Right? You have to get to minus 50 before you're able to get all or none. John?
You said the axon hillock is where it becomes minus 50.
Yes, yes. Once minus 50, it reaches, once the axon hillock detects that the cell membrane potential is at minus 50, that causes the action potential to trigger. So that's clear. Okay? So when the action potential goes all the way down to the axon terminal here, okay, it goes to this region in the presynaptic terminal here. Once it reaches the presynaptic terminal, okay, this causes the opening of a different kind of ion channel. And this ion channel I'm going to represent here. This causes the opening of calcium channels, which results in the movement of calcium into the axon terminal. Okay, mechanically gated channels detect some sort of sensory modality in the dendrites. Sodium rushes into the cell once it reaches minus 50 millivolts. Action potential happens, transmits action potential signal all the way down the axon, reaches the axon terminal, the point of the axon terminal known as the presynaptic terminal. Once that detects the action potential signal, it opens calcium channels. Calcium comes into the axon terminal. Does anyone know what happens when calcium floods into the cell at the axon terminal? Did you have another question? Sorry? Vesicle. So actually there's something in vesicles that's released from the axon terminal. Something is released from the axon terminal. Neurotransmitters, right. I'm trying to remember your name. Kayla. Yes. Okay. Good. So when once the action potential is transmitted down the axon, calcium channels open, and there are actually neurotransmitters that are in vesicles inside the axon terminal that are released into the synaptic cleft. Okay, I'll just draw a few neurotransmitters here. So these are neurotransmitters. Does anyone know any names of general neurotransmitters? Great, acetylcholine. So general, we could say acetylcholine. Okay. Another one we're going to talk about when we get to basal ganglia disorders. Another big one is going to be dopamine. Those two are general neurotransmitters. There's also, in addition to that, there's a lot of different neurotransmitters we could talk about. But does anyone know what the major excitatory neurotransmitter is in the CNS? It's an amino acid. Does that help? Probably not. Sorry. The major excitatory neurotransmitter, let's say major MAJOR, excitatory neurotransmitter is going to be glutamate. So major excitatory is going to be glutamate. And the major inhibitory that you need to remember, anybody know the major inhibitory? It's going to be GABA. G-A-B-A. Okay, and so if the cell, the first neuron sequence wanted to excite or cause depolarization, as Alex said, of the next neuron sequence, which neurotransmitter do you think it might want to release? An excitatory or inhibitory? Excitatory. Yeah, so it's probably going to release glutamate. If you want to depolarize the next neuron sequence, you're going to release glutamate. If you want to hyperpolarize it, you're going to release GABA. Yes? Yeah, that's your presynaptic terminal. Okay. Just the terminal, axon terminal, right in front of the synapse. Yes.
Just to clarify, hyperpolarized is GABA, glutamate is depolarized?
Glutamate is depolarized. Depolarized means the next neuron sequence will fire. You're going to excite it. And to hyperpolarize, if you use GABA, it'll bring it below threshold. Okay? Or way below resting membrane potential, hyperpolarized. So the chances of it firing or reaching threshold become much less. That makes sense. Now if we put the first graph together with this, does this help a little bit to see how the neurons work with action potentials? And this is going to be what you need to know. And what happens is the next neuron in sequence in these dendrites, they actually have receptors. So this is going to be in the dendrite here of the postsynaptic neuron, you're going to have a receptor that's going to be specific for the binding of a neurotransmitter. Do you guys have a question? No?
I was just wondering, like, if you're low dopamine and take anxiety pills for like dopamine, it doesn't go through the neurotransmitters because they're missing, correct? So where does it go?
So you have to go back. I missed the beginning. You said if you're taking what for?
Like medicine to get more dopamine, like if you are like depressed or something, since the neurotransmitters aren't there producing dopamine, where does it go? Like the medicine.
Where does it go? Yeah, so can you give me, so that's more of a pharmacology question, if Dr. Blair wants to answer that. So as far as, she's talking about taking anti-anxiety pills to increase dopamine. I mean, I know Cinemac, but that's a little bit different.
Well, so when your neuron releases the neurotransmitter, like you see in this picture, after the neurotransmitter gets released, it gets essentially vacuumed back up into the presynaptic terminal. What drugs for depression do is they do what's called inhibit the reuptake. They block the vacuum so that the dopamine or the norepinephrine or the serotonin stays in the synapse and keeps on stimulating that second neuron. So
Yeah.
it effectively increases the concentration of the neurotransmitter.
Yeah, so that's for serotonin, and that's for the dopamine. I don't know anything about anxiety medications and dopamine. That's where I was lost. That's okay. Are there anti-anxiety medications that are dopamine based? Okay, so I don't know that much about medications for anxiety or dopamine versus. I know SSRIs. That's the most common one, for depression overall. But does that answer your question?
Yeah.
All right. Sure. Well, that's Dr. Blair. Thank you. Any other questions? Okay, so what I was saying is on the postsynaptic dendrite here, you're going to have a receptor for the specific neurotransmitter, depending on what it was, and that's what I've drawn here. This is going to be a receptor for neurotransmitter. Okay, and that receptor for the neurotransmitter, if it binds neurotransmitter, starts another local potential, and this local potential is going to be a ligand, and the ligand is going to be the neurotransmitter. That's a ligand-gated channel. Okay, so for the receptor for the neurotransmitter activates another set of channels, known as a ligand-gated channel. The ligand is going to be the neurotransmitter. So the more neurotransmitter binds to the receptors that are available, you're going to start to increase sodium again in the cell. Okay, and as sodium starts to rise, once you get enough neurotransmitter release, once you reach minus 50, signal is transmitted into an action potential, which again is that voltage-gated ion channel. Make sense? So that's how the information goes from the environment through the neuron chain all the way back to the CNS. Questions? Okay, let's move on then. That's enough about action potentials and basics about neurons. Let's talk a little bit about neuron histology. Does anybody know what histology is? What's histology mean?
Tissue.
Tissue.
Study of the
Study of tissues.
body
But
tissues.
so in thinking about neuron, if I say histology, are these tissues so you're looking at your skin with your eyes, or how do you usually look at histological sections?
Microscope.
Through a microscope, right. So it's the microscopic view of what these things look like. Okay, does that make sense? So all we're going to do is look at various kinds of neurons and draw pictures of what they actually look like in a microscope. What I have here is the neuron histology is at the top. On the far left, I'm going to put the function of the neuron, okay, that we're going to draw. And down here on the bottom, we're going to draw the structure. So we're going to talk about two things. So what's the function? And then what does that look like? We're actually going to draw the structure of that particular neuron. I'll go way back to the first day of class and talk about sensory information. So if I say up here, this is going to be sensory neurons in general. So sensory, is that going to be afferent or efferent? Afferent, correct. So that sensory is afferent information coming into the system. Okay, sensory information coming into the system. We talked about a big category of sensory information, and that was somatosensory. So the function here is going to be, first category of sensory is going to be your somatosensory. That's information or sensory information coming from the body. That's your somatosensory. And the somatosensory neurons have a very specific shape to them. Okay, so the structure, what that shape is called is a pseudo, P-S-E-U-D-O, unipolar neuron.
How do you spell that?
P-S-E-U-D-O-U-N-I-P-O-L-A-R neuron. So a pseudo unipolar neuron. This is going to be the basic structure of neurons involved in the somatosensory system. Okay, they're called pseudo-unipolar neurons. And this is the guy, the pseudo-unipolar neuron is what we talked about in the PNS. The gray matter is located. Does anybody remember where the gray matter was located in the PNS? We talked about one specific structure. Something called the dorsal root ganglia. Okay, so the pseudo-unipolar neuron is the one that's going to reside in this dorsal root ganglia. And what it looks like is, go ahead and draw it in green. So you're going to have these dendrites here. Okay, and then below the dendrites you're going to have a stalk-like structure. And then sticking out, you're going to have another little stalk, and attached to that stalk you're going to have the cell body. There's the cell body with the nucleus. And you have another stalk-like structure. And finally you have the axon terminal. We'll go through and label those. So these are your dendrites here. Okay. This is your cell body. Okay. At the very bottom, you're going to have your axon terminal. Okay, so these are your first order somatosensory neurons that reside in your dorsal root ganglia. They look like this. The structure is known as a pseudo-unipolar neuron. Essentially you have your dendrites at one end, you have your axon terminal at the other end, and your cell body's in the middle, but it's sticking off of a stalk. So that's your basic somatosensory neuron. That's what it looks like under a microscope. Question, Alex?
Does that stalk have a name?
Not that I'm aware of, no. Okay. The other, in addition to the somatosensory sensory information, we also talked about other things like vision and olfaction. Vision and olfaction, neurons involved in visual system and olfactory system look a little bit different. They have a different name. They're called bipolar neurons. sensory system, use the pseudounipolar, vision and olfaction, use bipolar. And the bipolar neuron looks kind of like the pseudounipolar, where you have the dendrites on the top. Okay, you have a stalk-like structure. But here you don't have the cell body off another stalk. You have the cell body kind of right in the middle here with the nucleus. And then you have your axon with your axon terminal at the bottom. That's going to be the basic structure for your bipolar neuron. Your bipolar neuron, again, these are the dendrites. This is the cell body, and this is the axon terminal. Just looking at basic structure. If you looked at neurons involved in the visual system or the olfactory system under a microscope, this is what you'd see. You'd see your basic bipolar neurons. Questions about that? Okay. Now, moving on to the next category. In addition to the sensory or the afferent information, we're going to have motor information. So to separate these out and make it a little easier to see, I'm just going to put dots down the middle here. Okay, and I'm going to put motor. These are your basic motor neurons. And motor is going to be efferent. Very good. Okay. As far as your motor efferent neurons, they are called multipolar neurons. And the multipolar neurons are no different than my stuffy friend that I showed you earlier. Okay, that's going to be your basic neuronal structure, where you have your dendrites here. Okay, your cell body with the nucleus. And for your motor system, you're going to have a characteristically long axon. Okay? And then at the bottom here, you're going to have your axon terminal. So this is going to be, for the motor system, you should know this is a long axon. A multipolar neuron with a long axon. And again, these are your dendrites. Long axon, and this is your axon terminal. Okay, so the neurons involved in the motor system are going to be your multipolar neuron, which has, when I think of a neuron, it's the structure that I usually think about. Questions about that? Okay, let's go ahead and take a 10-minute break and regroup at 3:50. Did you have a question?
Which functions for the multipolar neuron?
Motor.
Just movement.
Yeah, movement. Yeah, motor system. Mm-hmm. Yeah, function is going to be just the motor system.
Real quick question.
Yeah, sure.
So you see the difference between cell body and cell.
Yes.
Does that mean it's at least different here? If that makes sense. Like I know that the helic was like right here, right?
Yeah, that's how I labeled it.
Like we don't really have to worry about that.
No, so I can tell you, when you think about it,
I get used to it.
start to make it but that right here
That would make more sense. I see it now because if that's the body, it's going to be beyond, right beyond the body into the axon.
I think I know I use this neuron. Let me turn it on.
I know I use that neuron just for simplicity.
Yeah.